Hi, this is Dr. Neela Bakore. In the previous segment, we tried to see some basics of photosynthesis, and we also started with light reaction. In light reaction, there are two possible pathways. One is non-cyclic photophosphorylation and the other is cyclic. We discussed non-cyclic one. Now, let us take the cyclic photophosphorylation. So, in this chapter of photosynthesis, now we are taking the cyclic photophosphorylation. In non-cyclic photophosphorylation, we saw that there were two photosystems which were involved, and the electron pair which is lost from the first center, that is reaction center of photosystem 2, does not come back to the same photosystem. That is why we called it non-cyclic. In cyclic photophosphorylation, there is only one photosystem which participates, and this photosystem has its reaction center chlorophyll. The photosystem which participates is PS1. That means the chlorophyll or the reaction center chlorophyll A is P700. So, here is chlorophyll A. This photosystem also has its primary electron acceptor. Now, the reaction starts the same way, that is the solar energy falls on these pigment molecules. Chlorophyll A absorbs 700 nanometers and all other pigments which are accessory pigments, they absorb different wavelengths according to the type of pigments they have or they are, and all these photons or energy which is collected by these accessory pigments is passed on to the main chlorophyll, that is reaction center chlorophyll. And because of this energy, a pair of electrons from this reaction center chlorophyll gets excited. Again, one pair of electron leaves the chlorophyll A molecule of photosystem 1. It is taken by the first electron acceptor, which is the primary electron acceptor of this particular photosystem. This pair of electron now passes through a chain of electron acceptors. The first one is ferredoxin. So, again let us write down the abbreviations and their full forms. FD is ferredoxin. After the primary electron acceptor, the electron would go to ferredoxin. From here and on all arrows, we need to write down what is moving from one acceptor to the other is a pair of electrons. Then it is taken by cytochrome complex. So, Cy is for cytochrome. In between this, there is one more electron acceptor which is plastoquinone. So, we will add that, or maybe just slight change here. Let us put plastoquinone here and then cytochrome. So, PQ is plastoquinone. And from cytochrome, it goes to another acceptor which is PC, plastocyanin. PC is plastocyanin, and this electron pair is given back to this chlorophyll molecule. So, the same pair moves through this chain of electron acceptors and it is coming back to chlorophyll A. Now, you know why the name is cyclic. So, reason, the pair of electron which is lost from chlorophyll A comes back to chlorophyll A. In case of non-cyclic, it was not coming back to it, and that is why photolysis of water was required. So, when water splits, the pair of electron which is released is given back to the chlorophyll, so that its loss can be made up. But here that photolysis is not required, because the electron pair is coming back to the same chlorophyll. Plus, when the electron pair moves from plastoquinone to cytochrome complex, at this particular place, energy is released, and this energy is used to convert ADP to ATP. There is inorganic phosphate also which is required, and here ATP is synthesized. So, in case of cyclic photophosphorylation, there is no need of photolysis of water, and only one thing is obtained, that is ATP. Whereas, in case of non-cyclic, we got ATP also and the reducing power, that is NADPH2 also. So, this is simple cyclic one. Now, let us see the differences between the non-cyclic and cyclic. Non-cyclic photophosphorylation and cyclic. First point, in case of non-cyclic, both the photosystems 1 as well as 2 participate. So, both photosystems 1 and 2 take part in non-cyclic. Whereas in cyclic, as we can see here, only one photosystem, that is only one, that is photosystem 1 participates. Second point, what do we gain out of these two cycles? In case of non-cyclic, the things which we obtain are ATP and the reducing power, that is NADPH2. Both are obtained. Whereas in case of the cyclic one, only ATP is synthesized. Or only ATP is obtained. In case of non-cyclic, because that lost pair of electron does not come back to the reaction center chlorophyll, photolysis of water takes place. So, in this, photolysis of water takes place. The reason why this is required is so that the pair of electrons lost from the first reaction center chlorophyll can be replaced. Here, as the electron pair comes back, no photolysis required. Another important thing, when photolysis takes place, along with electron and proton, oxygen is also given out. As in case of cyclic, no photolysis, so no oxygen. So, in case of non-cyclic, oxygen is evolved as a result of photolysis, whereas in case of cyclic, no evolution of oxygen. The non-cyclic photophosphorylation, it takes place in all normal or optimum condition. It takes place in optimum condition. That means carbon dioxide concentration, temperature, everything is optimum. whereas the cyclic one takes place when this NADPH2 accumulation is more. So, if its utilization is less and it keeps on accumulating, then as both these things are required for the dark reaction to take place, if only one thing is accumulating, the other thing which is required is ATP. So, in this cyclic one, as only ATP is synthesized, then in that situation only cyclic photophosphorylation will take place. So, this takes place under, it takes place when NADPH2 accumulates. So, in normal condition non-cyclic would take place, and that is why we say that most common photophosphorylation which is taking place is non-cyclic. But if conditions change, that means some product keeps on accumulating, then it shifts to cyclic photophosphorylation. So, it is a condition dependent cycle, whereas this would take place in all normal conditions. So, basic difference between cyclic and non-cyclic photophosphorylation. Along with the complete cycle, we can add couple of points more. For example, in case of cyclic, there is only one electron transport chain, whereas in case of non-cyclic there were two, one between photosystem 2 and photosystem 1, and the second one was photosystem 1 and NADPH2. So, we can write that also as an additional point. In case of non-cyclic, there are two electron transport chains which work, whereas in case of cyclic, one electron transport chain works. But the first 5 points are the most significant points. This is just to add the number and just to make the differences even more clear. So, important point, non-cyclic, both photosystems work, cyclic only one photosystem. In case of non-cyclic, two products are obtained, ATP and NADPH2, whereas in case of cyclic only ATP. Photolysis of water is required in non-cyclic to replace the electrons which are lost from the reaction center chlorophyll, as in cyclic the electrons come back to the same point from where they were lost. So, no photolysis required. Photolysis also results into evolution of oxygen. So, that is evolved in non-cyclic, no oxygen released in cyclic. Condition when non-cyclic would take place, all normal, optimum condition. Cyclic would take place only when NADPH2 accumulation is more, and then number of electron transport chain. So, this is light reaction. It can take place in two ways, a non-cyclic pathway and a cyclic pathway. And the reason we call it photophosphorylation is because it is light dependent, and phosphorylation is addition of phosphorus or ATP synthesis, which takes place in both non-cyclic as well as cyclic. Now, before we take up dark reaction, let us understand this part. We have written ATP synthesis. Actually, when we make this cycle, we just write ATP here, but ATP synthesis is not like this. It takes place by a specific mechanism which is called chemiosmosis. So, let us understand that first and then we will come to dark reaction. The chemiosmotic theory for ATP synthesis was given by a scientist Mitchell, and the theory is called Chemiosmotic theory of ATP synthesis. We are representing the complete thylakoid membrane in this diagram, so that we are able to understand each and everything how this ATP synthesis is taking place. This is the thylakoid membrane. The side is outside, this upper side. That means here there is stroma, and the side which is inside is the lumen. Just little explanation of what is happening in the thylakoid membrane. Light reaction takes place here because the membrane has the pigments. So, say this is PS2 and PS1. Here in this small area we are trying to represent what is happening during non-cyclic photophosphorylation. Now we will not write everything here, we will simply represent what happens. Here there is a reaction center chlorophyll, the electron jumps, it is taken by the primary electron acceptor. Then we made electron acceptors here, and this electron comes to the reaction center of PS1. From here it is again taken by the primary electron acceptor, and through one more acceptor it is finally taken by NADP, this pair of electron, and this NADP changes into NADPH2. This whole thing which is happening is non-cyclic photophosphorylation. In this case, when the electron pair moves through this electron transport chain and it comes to cytochrome area, that is the time when energy is released. So here energy is released. This energy is used to pump protons from the stroma into the lumen. So now this proton will come in to the lumen, and from where did it get energy? It got the energy from the electron moving from one electron acceptor to the next. It releases energy. There we showed that at cytochrome ADP gets converted into ATP. This is the place the energy is given out. So here the energy is released, and that energy is used to pump proton from stroma into the lumen. So now the lumen has proton. Similar thing is going to happen in case of cyclic photophosphorylation. Only difference is going to be that instead of two photosystems, there is going to be only one photosystem. So there also energy is released when it comes from plastoquinone to cytochrome. That energy will also be used to pump proton from stroma into the lumen. In the lumen, photolysis is also taking place. That means water is also splitting to produce 2 electrons, 2 protons and oxygen. So there are protons which are collecting in the lumen. This results into high proton concentration. Due to accumulation of protons, protons are coming from 2 areas. Which are these 2 areas? Proton accumulation is due to 1, protons are being pumped in from the stroma into the lumen due to pumping of protons from stroma to lumen with the help of energy. With the help of energy released from the electrons. So when the electrons move through this electron transport chain, and they release that extra energy, protons are pumped into the lumen. This is one. The second point why protons would be inside here, it is the protons produced due to photolysis. So there are two reasons why there are protons in the lumen. One, because of photolysis of water, and two, because of pumping in of protons which takes place with the help of energy which is released normally at cytochrome complex. Now, because of accumulation of protons, a gradient is created. Lower concentration of protons outside, higher concentration of protons inside. Plus, accumulation of protons changes the pH also. More H ions would make the pH acidic. So, because of these two things, these protons would tend to move back from their higher concentration to their lower concentration. This membrane cannot let protons go on its own. There are some special structures which are known as ATP complex. So, this structure is ATP complex. This ATP complex is made up of two parts. The part which is embedded in the membrane is known as CF0 particle. C is for coupling factor. So, this ATP complex has two coupling factors. One is C for coupling factor, F0. So, this one which is embedded in the membrane, this is CF0, and so this is an integral protein. Another part which is attached to it in the form of a head is a peripheral protein, and it is known as CF1. So, these two coupling factors, which are basically proteins, make the ATP complex. Protons can move back only through these ATP complexes. The F1 particle or CF1 particle acts as ATPase, the enzyme which is going to help in the synthesis of ATP. So, these protons would move back into the stroma through the same particles. So, when 3 protons go back, when they go back, according to the concentration gradient from higher concentration to lower concentration, they release the energy and go back into the stroma. This energy is utilized to combine ADP and inorganic phosphate and ATP, that is adenosine triphosphate, is synthesized. So, 3 protons when go back, the amount of energy released is sufficient to make this bond between the diphosphate and one more phosphorus, so that ATP can be synthesized. So, basically whenever we draw these cycles, the cyclic or non-cyclic photophosphorylation, we say at cytochrome complex energy is released, and that helps in synthesis of ATP. But it is not one step. This energy is used to pump protons inside the lumen from the stroma. This results into accumulation of protons in the lumen, increases the concentration of protons. Protons tend to move from higher concentration to lower concentration. This is possible only through ATP complex. This complex is nothing but made up of proteins, 2 proteins which are known as coupling factors, CF0 which is embedded in the membrane, and CF1 which is peripheral. So, when 3 protons go back to the stroma, the amount of energy released is utilized to synthesize 1 ATP. This process is known as chemiosmosis. So, chemiosmotic theory tells us how actually ATP is synthesized. So, next time when we draw these cyclic and non-cyclic photophosphorylation, we will draw it like this. Wherever we show that electrons move from the electron acceptor to cytochrome and then again to another one, and here energy is released. This energy is used to synthesize ATP. But we have to keep this thing in mind that ATP is not generated right there. ATP is used to create a concentration gradient, and when these protons move back, that is the time energy is synthesized, and for that we need ATP complex. So, this chemiosmotic theory given by Mitchell is a simple explanation of how we use this energy, or plants use this energy to synthesize ATP. So, during non-cyclic and cyclic, in both the cases ATP synthesis takes place, and this is the mechanism. So, now after this chemiosmotic theory, we have understood how ATP is synthesized. Now, let us talk about the dark reaction.